A recent study has found that gut bacteria plays a crucial role in helping T cells repair muscle. This exciting discovery highlights the importance of maintaining a healthy gut microbiome to support the body's natural healing process. No details were left out in the study, which suggests that the presence of specific gut bacteria can aid in muscle recovery. The findings are truly thrilling and emphasize the significance of optimizing our gut health. In mice, it has been discovered that regulatory T cells present in the colon migrate towards muscles and aid in wound healing, which has sparked curiosity regarding the potential impact of antibiotics on injury recovery. This discovery is quite exciting and raises many questions about the interplay between antibiotics and wound healing. Exciting research published on February 22 in Immunity suggests that trillions of bacteria residing in the gut play a critical role in tissue regeneration. The study found that T cells in mice's colon rely on gut microbes to fix muscle injuries. Without these beneficial microbes, inflammation could spiral out of control, hindering healing and causing fibrosis. It is clear that the gut's microbiome is essential to the body's healing process, and this discovery opens up new avenues for further research. According to Harvard Medical School immunologist Bola Hanna, the microbiota is playing a bigger role in influencing our immune system and overall health than previously thought. In his research of regulatory T cells, which act as the peacekeepers of the immune system by controlling inflammation, Hanna has found that the microbiota has a significant impact. This discovery is truly thrilling and highlights the crucial importance of understanding the relationship between microbiota and our health. Alexander Rudensky, an immunologist at Memorial Sloan Kettering Cancer Center, expressed great interest in the recent study's findings that the immune cell populations in the gut have a significant impact on physiological and pathophysiological processes throughout the body. He believes that this discovery opens up new avenues for exploring the many ways in which these cells generated in the intestine can affect various aspects of physiology. Hanna enthusiastically explains that the study began unexpectedly when he was investigating the significance of regulatory T cells in the process of wound healing. After an injury occurs, these cells rush towards the damaged muscle and their quantity reaches its maximum after 4 days, leading to a switch from an inflammatory to an anti-inflammatory state in the tissue. The transition, which is a pivotal stage in the process of wound healing, is believed to be facilitated by the regulatory T cells. No details were overlooked during the inception of this study. At the beginning of the research, Hanna used single-cell RNA sequencing to profile the T cells present at the site of injury. During the analysis, she discovered various types of T cells, but one captured her attention the most. Hanna noticed a particular kind of T cells that expressed a transcription factor named ROR gamma. This factor is typically found in regulatory T cells located in the colon. Hanna was thrilled with this discovery. The colon T cells have a significant responsibility of safeguarding the beneficial microbes that reside there and preventing any attack from the immune cells. Additionally, they also contribute to the metabolism and digestion process, which is truly remarkable. Hanna was fascinated by the initial discoveries and decided to prove that these cells actually traveled from the gut. He and his team utilized a procedure named optical tagging to monitor the movement of these cells. They genetically modified mice to express a unique type of green fluorescent protein all over their bodies, which transforms into red under the influence of light. The researchers then fired a laser at immune cells in the gut of the mice. Hanna expresses his excitement, saying, "We could now trace these colonic regulatory T cells and monitor whether they could travel from the colon to other locations." The team discovered that the cells didn't only migrate to the muscle but also to other parts of the body, such as organs. Moreover, the presence of tagged cells in the injured tissue indicated that colon cells had indeed traveled to the muscle after an injury. This exciting finding showcases the ability of these cells to travel throughout the body. The scientists were thrilled to discover that the T cells present in the gut and muscle of recovering mice were identical. To prove this, they employed T cell receptor (TCR) sequencing, which profiles T cell receptors. TCRs are distinct markers on the surface of mature T cells that attach to a particular antigen and are unique to a group of cells from the same parent, known as a clonal population. The findings demonstrated that the regulatory T cells in the gut and muscle during the healing process were part of the same clonal populations. This discovery further confirmed that these T cells originated in the gut and migrated to the muscle. Although the function of regulatory T cells in muscle repair was not well understood, researchers conducted an experiment using mice that lacked ROR gamma plus regulatory T cells, which resulted in slower healing and fibrosis. Through flow cytometry, the researchers discovered increased levels of IL-17, an inflammatory cytokine, in the wounds of these mice after an injury. Uncontrolled levels of IL-17 have been linked to delayed wound healing. The findings of this experiment have excited researchers. The research team discovered a direct link between T cells and gut microbes. They observed that mice raised in germ-free environments or treated with antibiotics did not recover as well from injuries and experienced fibrosis compared to normal mice. This is because regulatory T cells in the gut respond to antigens from food and bacteria, but remain inactive otherwise. In the absence of microbes, these regulatory T cells were absent in the colon and injured muscles of mice. The findings are thrilling and offer new insights into the connection between gut microbes and the immune system. Hannah expresses her excitement towards the discovery that gut bacteria have a significant impact on the process of wound healing. She believes this finding is fascinating and noteworthy. Excitingly, the team delved into the significance of gut-induced regulatory T cells in various healing processes. They discovered that these cells, which are activated by microbiota, not only aid in the recovery of muscles after an injury but also facilitate the healing of the liver from damage. In non-alcoholic fatty liver disease mouse models, regulatory T cells decelerated inflammation and relied on the microbiome for their functioning. Hannah is excited about the implications of the study's findings on the use of antibiotics in treating tissue injuries. The dependence of colon regulatory T cells on microbiota raises questions about the judicious use of antibiotics for patients just after surgery or with severe wounds. Moreover, the study's linkage of IL-17 to autoimmune disorders and tumorigenesis suggests that the gut may also influence other inflammatory disorders. Hannah finds the cross-communication between different parts of the body fascinating and believes that they have an impact on us in ways that we are yet to comprehend. Thanks for watching.